Dear colleagues, in partnership with the Österreichische Gesellschaft für Nephrologie, the Société Francophone de Nephrologie, Dialyse et Transplantation, and the Deutsche Gesellschaft für Nephrologie, Marcus at Home welcomes Professor Ole Frøbert in Örebro, with whom we would like to discuss immobility-associated thromboprotection, focusing upon a recent science publication from Dr. Frøbert and colleagues, which studied hibernating brown bears. So thanks a lot for being with us a second time. It's a pleasure to have you, Ole.
Thank you very much.
Thank you, Gunnar. Hello, everybody, and welcome to our today's program also from my side. Today, we will cover a clinically really relevant problem. It's venous thromboembolism. It's a fact that challenges us every single day in the clinic, and at least even made it to the headline news all over the world. And some of you may have watched the German Tagesschau on the 13th of April. Tagesschau still may be the most popular news program in Germany. And the show on the 13th of April, the show brought up the question how bears can help to understand thrombosis. And for all of you who may be wondering what bears have to do with thrombosis, it's all about a recent publication in Science with the title Immobility-associated thromboprotection is conserved across mammalian species from bear to human. And we are really happy to host one of the leading co-authors today. So welcome, Professor Ole Frøbert. Ole Frøbert is interventional cardiologist and professor of cardiology at Örebro University Hospital in Sweden, and is also affiliated with AU Health's Steno Diabetes Center in Aarhus in Denmark. And at this moment, let me also introduce our co-host today, Professor Florian Langer. He's an expert in the field of thrombosis and hemostasiology, and every time we need advice in that field and with this topic, we call Florian. He works at the Department of Internal Medicine and Oncology at the University Hospital in Hamburg-Eppendorf, and he's also a member of the board of the German Society of Thrombosis and Hemostasis Research. So welcome, Florian, again. So now, without any further delay, let me hand over to you, Ole, to give us an introduction to the topic and the results of the paper.
Thank you very much, Professor Custodis. So thank you, everybody, for inviting me to give this short intro to our study on thromboprevention in hibernating brown bears. This is a project that I have been chairing for 13 years together with ecologists and biologists in mid-Sweden. And the whole purpose has been all the way through to translate, to learn from bears to the benefit of humans. A little context. Brown bears hibernate, they lie still, almost completely still for 6 months. They have a very slow heart rate, very low flow condition, around 10 beats per minute. And they don't get thromboembolisms while they are hibernating. So we ask ourselves if inspiration from bears could inform a new type of thromboprevention in humans, since bears don't get bleeding when they avoid their embolisms. And there's a lot of in this project that is quite atypical. It's not conventional medical research, obviously, but also in most research that we do as doctors, we have a novel animal or organism. It could be a cell, or it could even be a human. And then we add some harm, and then we try to understand the pathology, and then perhaps we can invent the treatment. With the bear, it's the other way around. Here we have species that should encounter a number of diseases, but simply doesn't. And our question is, can we find the protective mechanisms, can we find the biomarkers, the pathways that protects bears from harm? And I will show you just a few pictures of how we do. These bears are wild, but they have a collar with a GPS transmitter so we can find them. Sometimes the bear is in a stone den, and then we send in David with a tranquilizing gun. Very few times the bear gets away in winter, but usually it's quite peaceful because bears in Sweden most of the time hibernate under an anthill or a fallen tree. And we just dig into the den, give the bear its awake when we have disturbed it, and then we sedate it with a dart, and then we take our samples. We take blood samples and muscle and skin and fat biopsies. In summer, we do it all again, but this time by helicopter. And again, because the bears are equipped with this color, they're easy to find. It's not very easy to get them out on an open spot where we can dot them, but the people that we do this research with are very, very skilled. And then we simply take the same samples once more. In quite a few bears, we have done echocardiography after we have sedated the bears both winter and summer. And you can see at the winter echo that it's really slow flow, and there is spontaneous echo contrast in all 4 chambers. This in a human would mean a very high likelihood of thromboembolism. In summer, the bear heart pumps like a human heart. It's a little bit more round, but there's nothing apart from that that's differs from humans. Also in winter, the ejection fraction is compromised in bears. So what we have in bears in winter, we have immobilization, and we know that increases coagulopathy. We probably have vascular stasis, simply because there is this extreme low flow and the blood must be queuing up to the heart. And it's also likely that we have vascular injuries simply by the fact that bears lie on very long periods in the same position and compress their capillaries and smaller vessels. So we have all the criteria for Werker's triad, but still, again, bears don't get blood clots. So in 2010, we looked at platelets. We thought, there could be something there. We did some very simple, basic measurements, aggregometry, where we tested bear blood in a standard aggregometer with various agonists. And we saw that during hibernation, aggregation or stickiness of the platelets is around half compared to humans. Of course, we can bring humans down to bear levels or below by giving them aspirin or clopidogrel, but still bears that do this spontaneously. Then we teamed up with some French colleagues, and they had access to blood from so-called Bedrest studies, which is done by several space agencies around the world. This was the French space agency. And then we compared, aggregometer findings in bears. It was a very uniform signal, low in winter and like in humans in summer. And it was a little bit up and down in these healthy volunteers. But overall, for the group, there wasn't any change. And that was only 3 weeks of Bedrest. And for a number of years, I tried to find a good platelet group. I asked several groups around the world if they would like to help us with the question and find out why is it that platelets don't aggregate as much. And in 2018, I managed at an ESC Congress in Munich to talk to researchers from Munich, Tobias Petzold, and his boss and the Matthias Mann group, very prominent mass spec people, and they agreed to go into this question. They came all the way from Munich with more than half of a ton of equipment to our field station and did a lot on site and a lot also back home in Munich. They found the same thing. Platelets do aggregate less in winter. But from mass spec, they also found that there was a protein, heat shock protein 47, that was 55 times higher in summer, and that had the same levels as humans in summer, and it was almost absent in winter. And then, of course, we asked ourselves, is this the explanation? It was the platelet membrane protein. In Munich, Petzold and his team talked to a center where they treat people that have encountered spinal cord injury and are paralyzed. And the doctors treating these patients told us that in the first month after patients have been paralyzed, there's a high risk of blood clots or thromboembolisms. But after a month, it's like the background population. And then we had luck. We found that the same protein is also lowered in these patients after 4 weeks. Also here we had help from a space agency, this time the German space agency and NASA, and they gave us blood from Bedrest studies, and we found the same. Again, the HSP47 was down after 4 weeks, so it had nothing to do with any kind of disease. Then we tested our hypothesis in pigs. In modern farming, when the sow gives birth to piglets, the sow is confined to a very small place and can't move for 3 to 4 weeks. And we found the same. And lastly, we did a knockout model of this protein in mice. And if the gene for HSP47 was missing, we couldn't, or almost couldn't, provoke thrombosis in a standard model in these mice. So that led to the publication of our findings in Science. There's a lot of work. Just that part took 1.5 years. And of course, it gave a lot of press for our research. That has not been easy over the years. Funding has been very, very difficult. The next step we will now try is to explore if this protein can serve as a biomarker in humans. So we have our discovery, and we are now applying for funding for a prospective study where we will have patients with deep vein thrombosis, also some with thrombus at the arterial side, myocardial infarction. And also we were in luck that a colleague that we have discussed with during the years, Alexander Choquer, he has access to blood samples taken at the International Space Station, and they are taken in such a way so we can also measure these proteins from these samples, so before, during, and after space travel. And then we will evaluate and see if we can refine this and make it into a standard biomarker, hopefully. And just to give a almost last picture of this bear model, when we as humans lie still for 6 months, we have, as mentioned, a risk of blood clots. We also have a higher risk of heart failure. We accelerate atherosclerosis if we don't move. We lose a lot of muscle depending on how still we lie. Osteoporosis is a problem. We burn all the fuels we have. We experience bed sores and much more. And the bear, on the other hand, avoids all this. And we are, of course, also trying to find explanations for some of the other observations, especially muscle is a hot topic in our groups right now. So thank you very much for listening.
Yeah, so thank you very much. That's really fascinating work. I enjoyed reading your article, and yeah, I've come up with a few questions. Unfortunately, they are focusing on thrombosis hemostasis because that's what I do in my daily practice, but I don't think that they're in any way any complicated. I'm just curious about your opinion. So when we look at the textbooks of thrombosis and hemostasis, they usually tell us that platelets are playing a role in arterial thrombosis, while the coagulation system is important for venous thrombosis. So were you surprised when you found out that the platelet compartment is responsible for the protective effect against VTE in your study?
Yeah, we were also, and coagulation and thrombosis is really not my strong side, but what I understood is that there is a very tight connection between this protein HSP47 and forming of these nets. Is it neutrophil
extracellular traps?
Traps, yes. Yeah, it's good. It's comfortable, comforting that you know much more about thrombosis. Yeah, so it's in the border area with inflammation, and perhaps that's a reason. We were also surprised that it was on the venous side and platelets, but as I understand, this area is expanding. So platelets are not that harmless on the venous side either.
Oh, I totally agree, because I think there's a recent clinical trial clearly indicating that in some certain trauma patients, you can actually give aspirin for thromboprotection against VTE, which is almost as efficacious as low molecular weight heparin. So there's certainly an overlap between the different systems, and it's not that easy that platelets are for arterial thrombosis and coagulation factors are for venous thrombosis. So that's very, very good confirmation of this major overlap between the different systems. So I understand that the reason for heat shock protein 47 downregulation in the platelet is actually less transcription of the messenger RNA in bone marrow megakaryocytes, correct?
Yes, that's what we think.
Yeah.
But we still don't know for sure, but it's probably
okay, because then it would suggest that this is actually regulated on the transcriptional level within the bone marrow. And my next question would be, but I guess that's only speculation. So what signals do you think are involved in this process? So how does immobility signal to the bone marrow to downregulate heat shock protein 47 production in the megakaryocytes? Do you have any idea or speculation on this?
No, in fact, we don't, but we have, of course, thought about it. If simply using your bones is that a stimulus for this? We will, next bear season, we will hopefully, because it's ethically hot, but hopefully we'll be able to have bone marrow biopsies from the bears, and that could help us to dig more into this question.
Okay, yeah, that would be very, very interesting to find out how this is regulated in the bone marrow microenvironment, probably. And also a question is, do you have any data on the time course of heat shock protein 47 downregulation, let's say after a human being gets immobilized? So is it starting immediately, and then you just have to look at the lifespan of platelets in the blood to see the effect, or does it take a while before this mechanism takes place? Do you know?
Again, we have an idea. We know if you go on a long journey on an airplane in economy class, you have this risk. So it's not very fast. With the bears, we are only allowed, and that's reasonable, we cannot disturb them more than once each winter. But the information we had from the doctors treating these spinal cord injury patients was quite informative, because they said their rule of thumb is 4 weeks. So 4 weeks, there's a high risk. After 4 weeks, it's like the background population. So it's probably around that.
Okay, because that's also a very, very frequent question in daily practice when a patient, let's say, has a stroke or spinal cord injury, and then the patient is in the hospital. There's certainly an increased risk of thrombosis, and then he goes to rehabilitation and maybe into a nursing home, and then the relatives ask us, so what about the heparin injections? So can we stop giving them? Does he still have an increased risk of thrombosis? And we usually say mobility itself is not a reason to give thromboprophylaxis. You always have to look if there's a trauma or surgery or infection. So your work is very good support of our clinical practice, that bedridden patients without any other problems like infections or trauma do not need thromboprophylaxis. So that's about it. Yeah.
Yeah, in the beginning.
Yeah, sure. But then later in the nursing home, or maybe during rehabilitation, when all the acute problems have vanished, and it's just the immobility that doesn't justify thromboprophylaxis with heparin, at least in our clinical practice. Well, that's it for now. Thank you very much. Very interesting again. Thank you.
Thank you.
May I ask a question regarding cardiology, a little apart from thrombosis? You showed us those beautiful echoes, and the heart on the left side to me looked some kind of like a patient with dilated cardiomyopathy, and on the right hand side during summer, totally normal. And I'm wondering, did you look at different morphologic features of those echo studies? Did you study those hearts and echo in detail?
Yes, we had two publications. The first was you could say the basic findings, and then the second one, more recent, we also went into all these very advanced echocardiographic features. And I'm invasive cardiologist, so I don't have them at hand, but it was we use all this spectral tracking, and what our echocardiographer for Peter Goss-Jørgensen concluded from this was that it's more the speed of the contraction that's affected. So ejection fraction is reduced, but it's especially speed. And his conclusion was that this has to do with calcium sensitization, that this is what is happening, but they don't have physical signs of heart failure. We did try to measure BNP, but some proteins we cannot measure with standard assays in bears, so we didn't succeed. But we have no reason to believe they have heart failure. But I agree, at the very first echo Peter did in winter, I showed it to a colleague after I came home, after I had been on call. I said, I did this echo last night. I said, do you think it's something we should proceed with? And he said, what? You should have informed me during the night. And then he saw the name of the patient was bear number 3.
Great. Another question I would like to bring up. You mentioned some hot ethical questions. Could you briefly introduce us? How is that regulated?
Yes, in Sweden, this is very strictly regulated, and that's good. So when we did this the first time, we only had a temporary permit, and now we've done this for 13 years, and we always, if we change anything, we have to apply again. Now they extended our permission, so we have, I think it's 5 years at a time now. The beginning was only 2 years, but we have data. We can show the bears, they do well, also in the long term. They breed and live long. But of course, this affects the bear. We disturb them, and therefore we are extremely careful. We don't want to harm them unnecessarily. But for example, to take a bone marrow sample, we will have to. Done some tests in the zoo, and now we will apply, and hopefully they will agree. We don't think there's a risk associated with that.
So I continue with the field of nephrology, because another very interesting feature in these animals, and you also published upon that, is that the bears become anuric during these 6 months. So without asking you to go into detail, but maybe in a few sentences, how do these bears manage to survive, apparently without urine production?
Yeah, their creatinine does go up, but it stabilizes around 200 to 250. And how they then take care of urea, there's only indirect evidence from bears, but from other hibernating animals, it seems like that's the gut microbiome that steps in and handles the urea in the gut, so we don't have very high levels of the byproducts. But we're still speculating.
And to put this into context, so the normal creatinine during summertime would be close to humans.
Yeah.
So 80 something micromoles. So it's kind of doubling then during hibernating.
Yeah, or tripling. Yeah.
Really?
But then it stabilizes. It doesn't go higher.
Yeah. And maybe then some more general questions. Why do these bears hibernate while polar bears and brown bears do not hibernate? So what's the reason behind it? Why do they do this?
I like your podcast, Gunnar, because you have to be a biologist, ecologist, nephrologist, whatever. The bear is interesting.
All of which you are, apparently.
Not really. But the brown bear is interesting because in Sweden, they do hibernate, but they also live in Croatia, for example, and they do rest for some weeks, but they don't hibernate for a long period. And also the bear is very unique among hibernators. It's one of the only animals where the temperature doesn't go down, the body temperature doesn't go down extremely. So in bears, it's between 32 and 34. And in most other hibernating species, it goes down to ambient temperature. There's even an Arctic ground squirrel where it goes down to minus. But that's completely different biology. When it comes to polar bears, they also have the ability, because female polar bears, when they give birth, they stay in a kind of a den with their cubs for some months, just like the brown bear, but only when they give birth. So it's something they can switch on and off, and they're very close, polar bears and brown bears genetically. So that makes it even more interesting. And there's no gene common to hibernators that separate or sets apart hibernators from non-hibernators. So it's on the protein level that all this happens.
So I will challenge you with another biological question. If anybody knows, surely, if bears in Scandinavia do hibernate, and if they don't do in Croatia, and now we also have bears in Austria and surely in Bavaria, does anybody know whether this may affect their lifespan, whether or not they are hibernating? Because surely this is such a strong change in their metabolism, in their physiology. So doing or not doing hibernation should maybe affect their overall lifespan.
Yeah, I'm not familiar with any data suggesting that. And we also know that hibernation is a response to the lack of food in nature. So that's the reason why they do it. And if you have a bear in a zoo in Sweden, some zoos think that they need to make water and food available to bears. And sometimes the bears wake up and take a little water and take a little food. It probably interrupts the hibernation biology. But again, the bear can choose this, apparently. And there's also, I don't know, I'm taking this too far, but there's also been speculations, of course, can we humans hibernate? And there was some newly published data from Neanderthals indicating from, they use bones to see how bones grew or developed, indicating that Neanderthals perhaps had long periods in winter where they more or less hibernated. But that's what we know on the human side. So perhaps.
What I found very interesting when reading your papers, you wrote that the mortality rate is much lower during hibernation in wintertime than in summer. I found in some of your publications mortality rate of 0.4 or something during hibernation, whereas it goes up to 58 or something during summertime. So apparently once the bears go to hibernation, they will survive to the next spring. And it's much harder to survive the summer than the winter, which was really very, very surprising to me.
It's probably because of us, because of humans. In Sweden, there is legal hunting. There's a quota for bears that can be hunted, and that's the end of August, beginning of September. It's not my favorite, and last year it was a lot of bears, almost more than 600. But that's a political compromise. And then there are traffic accidents, and then bears can be very tough to each other. So they also have fights. And it's not easy to be a female bear with cubs, because if the male bear that she had the cubs with is probably suddenly pushed out, a new male bear will come in and will challenge her and start with trying to simply kill the cubs, because these kids are not mine. So it's a rough life being a brown bear. And the few deaths, because we have data more than 30 years back in the den, were probably simply because another bear broke into the den and killed the bear inside. So apparently they just survive hibernation without problems.
And how do you find them in wintertime? So are they all marked in Scandinavia, each and every bear? Or how do you know where to go in order to take your blood samples?
We have around 50 to 60 bears in Sweden out of a total of 3,000 that have these GPS transmitters, but they also have a VHF transmitter. So we can see when they go into the den, we can see where the signal stops, and then we have an indication. And then in winter, we use old radio equipment to track them on the VHF transmitter. It's not that easy, but that's the way we find them.
So I would have 2 or 3 final medical questions again. Does anybody know how the bears manage to bring their heart rate down to this 8 or 10 beats per minute from, if I got it right, 90 during summer times? So it is mediated by hormones, or how did this happen?
Again, we don't know, but we know that the entire metabolism of the bears is reduced to 25% of normal. So it makes sense that the bear heart goes down with the rest of the body, but it's still astonishing. And the reason why we know it for a fact is that we had these small loop recorders in 30 bears in total. So we know for sure that this is really their heart rate. It goes down as far as 8 beats. And we also published a case report on this, where we had one bear who had, and this bear had the same name as me. Unfortunately, it was shot later, but it had a pause in heartbeat of almost 50 seconds. And they have asystole quite often for long periods, but 50 seconds, no heartbeat.
And then you told us that this hibernation in bears is not so profound as in some other animals. And I like the term you used in some of your papers. You called them shallow hibernators, which sounds very nice, actually. Wouldn't it be easier to go for smaller animals who have even more profound hibernation, who are surely much less difficult to find in wildlife, to do such science? Are you aware of anybody doing the same kind of research as you do in smaller mammals?
Yeah, there's a lot of research on smaller animals, especially these ground squirrels. But the reason that we stick to bears is really the body temperature. The smaller ones, they go down to very, very low temperatures, maybe for 10 days, and then they go up for a few hours to normal temperature and down again. Our interpretation is that this is a completely different biology. And it's possible that you can translate for this, but we think that the bears, it's a much better model to use as a translator.
So my last question, you not only found heat shock protein 47, you also found a couple of other proteins of which I never heard before, like SYK and SAP1 and ERB44. Will you or will the Munich team also try to better identify these, or will the next years rather see the focus upon heat shock protein 47 because it's, as far as I understood it, the one protein with the most tremendous changes?
We don't ignore the other findings, but a way to illustrate how lucky we were with this heat shock protein, we also found another protein that was almost 50 times upregulated in winter, and that's sex hormone binding globulin. in humans, we think it's only a transport molecule for sex hormones. And we had a PhD student, she used 3 years of her life studying this and found in a very short word description, she found nothing. So we were in luck with this. We're not sure we have the complete study on sex hormone binding globulin, but it just illustrates that it's not every signal that obviously leads to breakthrough.
Okay. Thank you. Professor Ole Frøbert joined us today, and we learned a lot about thrombosis and hemophysiology in bears. Maybe we can find the term from we learned a lot about from bear to bedside today. And thanks for coming. Thanks for having you. Bye-bye.
Thank you very much.